Hello and welcome. So the very first thing that I wanted to talk about before I get into what you need to do today is that many of you have missing work, missing assignments that you need to get done. Right now they're in the gradebook as zeros, but when you put in the effort and do the work, I will regrade them and put in the appropriate grade. But if you do not put in the effort and do not do the work, then the zero stands. Your grade is your responsibility, so let's make sure that you are getting your work done every day. Should be done consistently. Right now, for many of you, it's piled up because you've ignored school for quite a few days or weeks. So it needs to start getting done. Today, though, is our final lecture before fall break, and we talked yesterday about biotechnology, the different fields of it, so the agriculture, sustainability, and the medical field, right? What biotechnology can do for that. And we talked about how biotechnology is a really broad category of science, how it's defined as a toolbox that solves problems, and while we talked about how that was a terrible definition for the word and how it really didn't describe anything, it's appropriate because biotechnology is so diverse, so different, has very many different fields and many different applications, so it almost needs that broad of a definition. Today, however, though, before we go any further into discovering more about biotechnology, we need to learn about DNA. So today's lecture is all about DNA, or deoxyribonucleic acid. We'll go into our activities, see what we're going to do today. You don't have any notes in your notebook for this. This is all computer-based activity. So one can't talk about biotechnology or a toolbox for solving problems without first understanding DNA and protein synthesis, or the building of proteins. So you're going to watch the following video. Can't remember how long this one is, maybe 5 minutes. After the video, you're going to answer the following questions. What are proteins made of? What do proteins make up? And DNA is thought as a recipe for organisms. Explain why we say this. Use knowledge from the video to help you answer this question. You're going to copy those and you're going to put your answers in a text box. Next, you're going to watch a video on base pairs. Did you know that DNA is a double-stranded, right? So it's not just one strand as two. Yep, Watson and Crick discovered this with data from Rosalind Franklin. We'll talk more about how maybe Watson and Crick don't deserve as much credit or the Nobel Prizes that they got, and how Rosalind Franklin really did most of the muscle work. But that's not a topic for today. Today we're just going to talk about the specific way that the DNA strands line up. Okay, so after watching the video, I want you to match the following nitrogenous bases with their base pairs: adenine, cytosine, guanine, and thymine. Match those up, which goes with which. You'll learn that from that video. And then finally, to summarize, I'd like you to watch a crash course video to really set all this DNA learning together. So crash course is a little bit long. I think it's about 8 minutes. I would like you to, for question number 5, give me 5 facts that you learned from this video. You're going to type your answers into the question, or you're going to type your answers to these questions labeled with the numbers into the submission box for today's assignment. Here is an optional part of your day. This is what we're going to be doing in class, and if you have the supplies, you're welcome to do it as well. I gave you a little video, and it looks like I put the wrong video in there, but I will fix that. No, that video is just not supposed to be there. Okay, so maybe it is, because that's, nope. No, it's not. That video is not supposed to be there. I will put the correct link. It's going to show you how to make the bracelet. So we're going to be making a DNA creation activity. If you have the supplies, you're welcome to do this at home. This part is optional. Don't have to do it. You're going to create a DNA bracelet. You'll need string, beads, and scissors. I'll put up the page that has the directions, but here's the procedure. So assign each bead color a DNA-based letter in the video, which I will put there, or the video is right here. Eleanor assigns her blue beads as adenine, her orange as cytosine, pink beads as thymine, and her purple beads as guanine. You're going to separate your beads into different plates or containers sorted by color. You can even label them if you like. Step 3, which combos which. You're going to tie a couple of knots about 2 inches from one end of your string, of your 2 strings. So you're going to take 2 strings, or take a really long string, fold it on half of each itself so you have 2, put a little knot at the one end where it's connected, and that's where you're going to start. You're going to choose a section of DNA options listed above. There's a list right here. This is the pattern you'll follow as you string the beads onto your bracelet. So you can pick any DNA you like. Here's the sheet for you. You can do chimpanzee, brown trout, a human, butterfly, spitting cobra, flesh-eating microbe, sweet orange, carnivorous plant, giant Madagascar, hissing cockroach, or the corpse flower. So those are all options for you to do. What you're going to do is you're going to take your beads and you're going to put them on in the appropriate order. So if I was doing the hissing cockroach, I would put whatever colors for G, whatever colors for A, whatever colors for T, T, C, G, and so forth. And then on one side of the string, remember you have two, so that's one side. And then on the next side, you need to line up the appropriate base pairs, right? So if I was doing this and I had my G on one side, I would put a C on the other, whatever color bead that is. With my A, I'd put a T. With my T's, I would put two A's. G, C, you get the pattern here. Hopefully this is going to help you understand more about the double paired structure of DNA, but also the fact that it has complementary base pairs. So we just went through step 6 and step 7. At the very end, you're going to tie a knot around the last bead of each string and then tie the ends of the string together. Lastly, you'll just twist the two strings together so the correct beads match up and then wrap a piece of thread around the two strands in order to keep them in the twisted position. There's a video that walks you through this. Again, that part is optional. What is not optional, though, is answering those 5 questions using the videos. So have a great day, guys. I will fix this, and I will see you after fall break. Again, if you have missing work, today is the day to complete that. Got it? Your grade is in your own hands. Have a great day, guys.